Instant Neuro! Brain vesicles. At the third week of development, the cephalic end of the neural tube shows 3 dilations. These are known as the primary brain vesicles. These are the prosencephalon, mesencephalon, and the rhombencephalon. By the 5th week of development, 5 secondary vesicles are visible. These are the telencephalon, diencephalon, mesencephalon, metencephalon, and the myelencephalon. Each of these vesicles gives rise to specific central nervous system structures and spaces. Rapid growth folds the neural tube, forming 3 brain flexures. These are the cervical flexure between the brainstem and spinal cord, the cephalic flexure, which pushes the mesencephalon upwards, and the pontine flexure, which generates the 4th ventricle. The cavity within the neural tube will form the contiguous space of the ventricles of the brain and central canal of the spinal cord. This space is filled initially with amniotic fluid, and then later with cerebrospinal fluid. As development proceeds, the telencephalon swells and grows posteriorly and laterally to create the cerebral hemispheres that envelope the thalamus and hypothalamus of the diencephalon. The basal part of the wall of the telencephalon thickens to form the basal ganglia. The mesencephalon, or midbrain, differentiates into the tectum and the tegmentum. The CSF-filled space at the core of the midbrain is the cerebral aqueduct. The rostral rhombencephalon, or metencephalon, differentiates into the cerebellum and pons. The cerebellum is formed by the growth and fusion of the rhombic lips. The CSF-filled space at the core of the metencephalon is the 4th ventricle. The caudal rhombencephalon, or myelencephalon, differentiates into the medulla. The medullary pyramids are bundles of axons coursing caudally towards the spinal cord. The CSF-filled space at the core of the medulla is the 4th ventricle. Instant Neuro!